July 26, 2007 here at the University of Baja in Ensenada. This beautiful university here at Baja, on the Baja Peninsula in the Ensenada area, it sits right on the bluff. It just, I mean, literally the ocean is right here, the Pacific Ocean. And we're going to head in here now to the to one of the lab buildings. Well, Oscar and Julio, really, we appreciate the work you guys are doing here for us, okay?
Appreciate you having us.
This is basically where you do the cultures. And
Jerry, how many cultures now? I see a lot of bubbles there, a lot of bottles, right?
there's only one. We're doing...
You mean this is what you do with one culture?
Well, this is the CX. They're doing all kinds of modifications on it, different types of buffers. We're trying to determine exactly what it likes and what it doesn't like.
Phase 1. Cryopreserved frozen microalgae strains are transferred to Petri dishes by inoculation using aseptic sterile methods. Phase 2. From Petri dishes, microalgae is inoculated in the liquid culture test tubes until a biomass density is sufficient to transfer into larger culture vessels. Phase 3. Further cell density of microalgae determines the transferring of cultures in the larger vessels for algae pond inoculations. These large bioreactors are currently being used for multi-strain testing. However, these bioreactors will further be utilized as microalgae inoculators for BTR's large-scale commercial 100-acre and 1,000-acre production facilities in 2008. After the harvest of algae and the bioreactors takes place, it has to be dried and the oil extracted by several laboratory processes. The dried algae is analyzed to determine its oil and protein content, carbohydrates, etc. The large bulk samples of algae from the Montana demonstration facility are also being analyzed here at the University in Baja. The CX13 strain grown at the Montana facility has shown excellent survival ability under extreme variances of environmental conditions such as temperature, pH, salinity, etc. The CX13 strain, after testing, has exhibited high growth rates, high protein content, and oil yield.
Okay, yeah, there's the gas chromatograph. What's Martha studying right now?
She's counting cells. Do different treatments on algae to see if they can grow faster pace.
This is one of the main studies you've done so far
to find out which algae was best.
Note, previous research on several strains of microalgae were tested prior to sending final strain CX13 to Montana.
Ciao. Okay, I'm just following Oscar and Julio into one of the main labs here. Additional cell capacity to produce a higher form of lipid. Will that then be an algae strain that we will go out into the ponds with?
Yes.
That's what we want to hear. We will then take the yield up, who knows?
Exactly.
On the same production, same rotation, same
requirements.
Everything has been done in Montana, can be duplicated, but we're now bringing a very high yielding culture then. You said something there. You said for commercial purposes now, right? And that's the whole goal here, right, guys? Yep. Now we're proposing a 20-acre, 100-acre, 1,000 acres, and then we're off. Great job, guys. Thanks a lot.
This is the Biotech Research Microalgae Demonstration Facility in Montana. It is one of the largest algae demonstration facilities in the world, with a capacity of 40,000 liters, providing valuable research data since April 2007, including environmental control of critical parameters affecting algae growth. The Montana facility has provided a giant step toward commercial production. The quantity of algae harvested in a 2-month period can almost fill the bed of this full-size pickup truck. The microalgae sample will now be transported to BTR's Research Center at the University of Baja in Ensenada, Mexico. Part of this sample will also be sent to other research facilities in the U.S. for joint testing of the oil yield content. This will all lead into BTR's commercialization program for the planned construction of a 100-acre and a 1,000-acre pond system in 2008.